Agi Erfane. It's online.
Good evening.
Oh, good evening.
Good evening,
Okay,
dear professors.
Sure.
Good evening, my dear professors. I'm honored to be here to present my research project on prognostic and predictive biomarkers in human cancer. Since this topic is very close to my background studies, please let me start with my master project, and then I'll start and then I will present my topic research. My thesis was about preparation of biocompatible nano bubbles carrying quercetin and their inhibitory effects on non-small cell lung carcinoma. During my master project, I mastered skills on MTTSA, PCR, real-time cell culture, and nanoparticle synthesis. And also, I worked on my cancer patients and gained valuable experiences. On the other hand, I participating in the course offered by Green Department in Italy, in Palermo, about nanovesicles. I gained a deep understanding of nanovesicles and their synthesis and their potential applications in drug delivery system, and including their design, characterization, diagnostics, and therapeutic treatments. And in the last 2 years, I studied and I researched about precision medicine, biomarkers, molecular, and kind of nanoparticle synthesis, and I published. I had 3 published about nanovesicles in colorectal cancer, and the other nanobubbles in precision and molecular medicine for cancer treatment, and the last one was about convergence of nanoparticles, artificial intelligence, and precision medicine in lung cancer. I wanted to talk about my project topic. This PhD project in cancer biomarkers aimed to identify how specific biomarkers are linked to patient outcome like survival or treatment response. This research will contribute to advancement in cancer diagnosis, treatment selection, and ultimately improve patient outcomes. Let's talk about biomarkers. A biomarker, short for a biological marker, is a measurable indicator of some biological state or condition, and biomarkers are used for diagnosing disease, predicting disease, and they can be found on various biological samples, such as blood or bodily fluids and tissue, and etc. About the types of biomarkers, diagnosis biomarkers used for detect or confirm the presence of disease For example, PSA on prostate cancer, and another biomarkers is prognostic. They are provide information about the likely course of a disease like HER2 on breast cancer. And we have another one, predictive biomarkers predict the likely response of a disease to a particular treatment, like cross mutations in colorectal cancer cells. Pharmacodynamic biomarkers indicate biological response to a treatment, like blood glucose in diabetes treatment. And the last one of the biomarkers, safety biomarkers, they use to assess the risk of toxicity or adverse effects of a treatment, like liver enzymes as a marker of liver damage. I have examples of biomarkers in cancer cells like BRCA2 or BRCA1. They found on breast cancer and ovarian cancer, and the other one, CA125, also in ovarian cancer, AFP, a marker for liver cancer and germ cell tumors, EGFR, and last one, PDL1. About the methodology, marker selection, we have to studies about previous studies, databases, and clinical trailers, and validation, about the validation, analytical validation, testing the biomarker for accuracy, precision, sensitivity, and specificity. About the clinical validation, accessing the biomarker's performance in predicting clinical outcomes in cancer patients. Sample collection and processing, sample size for statistical significance, and sample sources about like blood, tissue biopsies, and about sample processing and storing samples to ensure integrity and consistency. Experiment methods, genomic analysis, like NGS, whole genome sequencing, or RNA sequencing, promote analysis, methods like mass spectroscopy, Western blotting, or ELISA, and metabolomic analysis using techniques such as NMR spectroscopy or mass spectroscopy to study metabolisms. Data analysis in bioinformatics tools like Python, Bioconductor, and the other. And about statistics analysis, outline the statistical methods for data analysis, including survival analysis, Cox regression, and ROC curve analysis. Predictive modeling, model of development, include steps like feature selection, training, and validation, and about model evaluation, such as accuracy, sensitivity, specificity, and AUC. About the expected results in this project, identification of biomarker association. Our research aims to contribution between specific moleculars like biomarkers and how patients fare with cancer, such as survival or response to treatment. Development of prognostic models. If I choose biomarkers linked to the patient prognosis, we hope to build models that predict the risk of cancer occurrence or progression. Evaluation of predictive potential will also explore if these biomarkers can predict which patients respond best to specific therapies. This could help tailor treatments for individual patients. And the last one, contribution to knowledge. This project aims to advance cancer biomarkers research. By understanding these specific biomarkers, we can improve our grasp of cancer and develop new diagnosis tools and treatment. And the final, I'm very excited about the possibility of joining Urbino University and contributing to the prognostic and predictive biomarkers in human cancer project. Thank you for your attention.
Thank you. This is a very clear review, but I can't see your projects. I mean, which cancer you want to explore, for example, how many samples, because I've seen, another view, but I don't see your projects in these 3 years.
I will choose lung cancer because of my master project was about lung cancer, and I specifically studied about that, and my point will about lung cancer and treat these people, and I want to join nanoparticles and with biomarkers for lung cancer.
Okay, thank you. Other questions from the board? No?
I may ask a curiosity. If in this project that you presented all the information, you talked about some statistical analysis, and if you don't know how many patients, how do you want to treat them, how are you going to manage it?
I think it's very difficult to answer because there isn't a clear project.
Yes.
It's fine. Don't worry. If you don't have an answer, it's fine.
Okay, thank you so much.
It's only if you I wanted to ask it because I thought that you had something in mind, but you presented something in general, so it's fine. Don't
Okay.
worry. Okay.
Okay. Thank you so much.
Thank you. Thank you so much.
Thank you.
Okay.
Thank you so much. Shafiq, hi.
Yes. Can you hear me now?
Hi.
So before starting and going into the main project that I have designed, just give me a brief introduction of me, as I know that you have my CV just a bit. I have done masters in microbiology from Comsat University in Islamabad, and the project that I have done in my masters is specifically the characterization, synthesis, and antimicrobial, antioxidant, and anti-cancerous activity of some of the metallic nanoparticles, which are effectively good against the lung cancer cell lines. So I have a previous experience of working on cancer cell lines, HUH7 cancer cell lines. So that was the major motivation from my side to apply in this specific project and the specific PhD. So now moving on to these are some of my previous study results. As you can see, there are some MTT assay results on the bottom left of your screen. So I'm not going into that because I have a limited time. So there it is. The project of priority theme will be the progress by active biomarkers in human cancers, and my just give me a second. So then the research project is basically investigating the molecular targets in transforming growth factor beta signaling pathway, which can result into evaluating novel therapeutic agents in head and neck cancer. So the major theme of this research is to investigate the molecular targets within the transforming growth factor beta signaling pathway, and which will evaluate some novel therapeutic techniques as well. The study is to go to to understand the basic role of TGF beta pathway in tumor progression, basically. So we can identify some potential genetic mutations as well, and at the end we will use some knockout techniques using CRISPR-Cas9 and some CAR-T cancer cell therapy. So moving into my research project, as you can see, this I have chosen the head and neck cancer. Why I have chosen this specific cancer is because the 90% of the cases are from the squamous cell carcinoma, and the 10% cases are from the non-squamous cell carcinoma. So this was the one major reason behind of selecting this head and neck cancer. And the prevalence is very high, as from the different literature that are present in the research engines, that the prevalence is very high. Every year, 6,000 new cancer cases, while more than 350,000 deaths, and the seventh most prevalent cancer in the world. And all these prevalences, so this is the major reason as well for choosing the head and neck cancer. These are some of the symptoms and some of the risk factors that are involved for causing this head and neck cancer. So it is not very much important, but the major thing for selecting TGF beta, why we have selected the TGF beta, the transforming growth factor beta. So we have to evaluate about the role of TGF beta signaling in cancer progression. Why? Because the TGF beta has a very complex and multifunctional pathway, because it has a dual role, as you can suggest from the literature as well. It has a dual role. It can act as a tumor suppressor agent as well as or as a tumor repressor agent. So it depends on the condition, on the stage of the cancer, and the promoter, how it will act as a tumor suppressor agent or a tumor progressing agent. So we have to evaluate that. It has a very complex structure. The transforming growth factor beta signaling pathway, it has a very complex structure. We will discuss about that structure as well. As I already explained, the dual role of the pathway, tumor suppression and the tumor progression. If it is acting as a tumor suppressor role, it can diagnose it can be used for the early diagnosis of cancer. It inhibits the cell proliferation. It can induce the cell apoptosis as well, and it can also regulate the tissue homeostasis if it is acting as a tumor suppressor role. If it is acting as a tumor promoter role, it can activate the epithelial transition EMT, which is called, in which epithelial cell lose the cell and cell adhesion, and it also loses the invasive properties. So we have to evaluate that, how the TGF pathway will act as a tumor suppressor or a tumor progression region. So as I already said, that it has a complex structure. It has a complex structure, and due to that complex structure, it has a ligand, it has receptors, it has some proteins as well, as I have mentioned in my fourth point. As you can see here, the SMAD2 and 3, when the signaling cascade will happen, During that signaling cascade, the ligand will bind to the TGF beta ligand, and it will bind to the TR2, which will phosphorylate the TR1, and then it will eventually goes towards the SMAD2, which join the 3, and then it will eventually activate the SMAD4. So we will also discuss about the TGF beta inhibitors, as few of them are mentioned here, and the role of microRNAs and SMAD7. So these are some points that why we have chosen transforming growth factor beta. So what will be the innovative aspects and possible advances? At the end, we will be a step closer to identify some new therapeutic agents, some predicting innovative, also broader application for other cancer cell lines. Because after this, if we are succeed to perform this research specifically, we can open the new gateways for treating other cancer as well. If we treat this head and neck cancer by this approach, what are the main objectives of the study? We will discuss about the few main objectives of the study. Number one objective is that the expression profiling of the TGF beta pathway genes and microRNA. We will choose the samples from the 200 family members. Second objective will be the correlation with clinical parameters and influence on the cell lines. I will go towards the cell lines after when we will discuss the methodology, but this is the second objective. After that, the third objective will be the screening of specific mutations in targeted genes. If we find some targeted gene, if we find some mutations, we have to screen that during the analysis of those targeted genes that we are analyzing. And at the start, I have mentioned that we will use some knockout models to get our desired results. We can use the generation in vitro CRISPR-Cas9 knockout models as well, some pathway inhibitors, and we can also use CAR T cell therapy for TGF beta modulation. So these are the main objectives of our research. Now, moving towards the methodology, I think this methodology will clear a lot of questions if they are in the mind. First of all, the first part will be the ethical approval. We have to take out we have to get the consent and the ethical approval for conducting this research. After that, we have to go towards the sample collection. While going towards the sample collection, there should be some concerns and some parameters while selecting those samples that the patients or the individuals from which we are taking the samples, either we are taking have specific growth factors or not. Some we have to evaluate the sample. We have to evaluate certain factors before concluding our samples. After that, from those samples, we have to perform some RNA extraction and see DNA synthesis. For RNA extraction, we can use different methodologies. We can use different certification techniques, and then we have to perform the cell lysis. And after that, we will get RNA precipitation and then RNA restoration. And after restoring that RNA, we will go towards the cDNA synthesis, and we will take that initial sample of the RNA, and then take that RNA sample, and then we can use the hexamer pairs as well, and the reverse transcriptase, and at the end we will use the PCR to get the cDNA synthesis. After that, we will use the real-time PCR. Why we are using this real-time PCR right now? Because we have to perform the genetic profiling, and for the genetic profiling, we have to use this real-time PCR to examine the expression profiles of TGF transforming growth factor beta signaling pathways. For that purpose, we will use the real-time PCR. After that, the major thing of our research will be some cancer cell lines, getting those cancer cell lines. It is, we can have the established cell lines, for example, SCC4, SCC9, SCC25, which are the established cancer cell lines, which are normally available in the market. We can use that cell lines, or we can use, we can culture some cell lines. We can also use primary cell lines, which are derived from the samples. After that, we have to perform another analysis, which is known as immunohistochemistry, which is used to analyze expression, localization, and distribution of specific proteins. So for that purpose, we can use the immunohistochemistry analysis. After that, we will use the PCR-SSR, which is specifically used to detect the genetic mutation or polymorphism, or we can also use for the detection of some point mutation. If there is some mutation is present in our samples, we can use this PCR technique to get them out, or to evaluate that, or to observe that. After that, we have to perform our CRISPR-Cas9 knockout models. We can use the CRISPR-Cas9 Cas9 technique to knock out the specific gene which will eventually regulate our TGF pathway for the tumor suppression. After that, we can use the TGF beta pathway inhibitors. Why we are using these inhibitors and what type of inhibitors there are? We can use the small molecule inhibitors, monoclonal antibodies, some ligand decoys, and some antisense oligonucleotides. We can use these type of TGF beta pathway inhibitors. At the end, we have a CAR T cell therapy. What the CAR T cell therapy is? It is basically chimeric antigen receptor T cell therapy that will involve the immunotherapy that involves the genetically modified T cells, and CAR receptors will bind to the specific T cells and give those T cell an effective target against those cancer cell lines or the cancer cells. So we can use the CAR T cell therapy at the end to get the desired results. So this is the basic methodology that I have planned for my research project. After that, what will be the expected results? Better understanding of the TGF beta pathway role in the head and neck cancer, identification of some elements contributing to the variant pathway, also the insight into the earlier diagnostic and better treatment strategies. As I mentioned that the TGF signaling pathway can, if that role is specifically to suppress the tumor cells, we can also use it for the early diagnosis of our head and neck cancer. So these are the expected results. This is the time frame of my research, how I have planned this research in the 3 years period. So first of all, month 1 to 8, collection of the samples, RNA extraction, DNA synthesis, expression of profiling of TGF beta pathway genes, and miRNA family members using real-time PCR. This is all the plan I have made to complete out my result, and at the end I have data analysis and interpretation using different type of statistic models, specifically use ANOVA as well, t-test and chi-square test as well. So this is my plan for the 3 years. So these are the references, bibliography that I have took some help to generate this research topic. Thank you very much.
Thank you so much, Tayab. Another question from the board?
Yes, I have.
Yes, sure.
Related to your previous expertise, how do you think to contribute in this project?
Yes, as I mentioned earlier, that I have some expertise of doing my research on the specific lung cancer cell lines. So from that expertise, from that research expertise, I can use some all of these molecular biology techniques, which can be RNA synthesis, real-time PCR, cDNA synthesis, cancer cell line handling, all of that stuff that I've done earlier as well.
Okay.
That I
so related to microRNA, for example, since you didn't show it in the methodology, how do you plan to analyze them?
MicroRNA analysis?
Yes, the microRNA 200 family that you indicated at the end.
Yes, basically the microRNA analysis, we have to perform that at the cell level. So we have to do the sample collection and preparation. After that, we have to preserve that RNA, and then we have to perform that RNA extraction. After
yes.
the RNA,
how do you make this extraction?
You are asking about which type of equipment I will use, right?
Yes, the methodology, yes.
Yeah, methodology. Okay. For RNA extraction, I can use different isolation kits. I can use microRNA specific isolation kits that are micro.
Sorry, try to speak slowly because it's quite difficult to understand you. Okay.
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 So I was saying that for RNA extraction, I can use different type of isolation kits, which can be specified mRNA isolation kits for the total RNA extraction, and for the quality check or the RNA quality check, I can use nanodraw spectrophotometer and the bioanalyzer of some kind. And
okay. Total RNA is fine, and then what are you going to use as technique?
Quality qPCR basically at the end.
qPCR, okay.
Yeah.
Okay, that's fine for me. Thank you.
Thank you very much.
Thank you, everyone that are still online.
Thank you, Professor.
Goodbye.
Thank you, all of you. Bye.
Bye.
Bye-bye.
Bye.
Bye. Bye. Bye.